Hey guys, so here we have another example of cell communication. We've already talked about the endocrine system, right? Which is that system, it releases hormones that then have a signal transduction pathway, right? Now we're at the nervous system. So this is going to be how neurons, which are these weird shaped cells, interact with each other. And so here I have a model of a neuron. You don't need to know the details of this diagram. You should know the general ideas of how the nervous system works. So information is generally going to travel in this direction down the neuron. You have the axon and the dendrites, right? So what that is, those are the parts that communicate with each other. Again, you don't need to really know these terms, but you should kind of understand the main concept, right? And what happens is you have neurotransmitters, little molecules that are going to be exchanged between here. So we have these are like branches. They branch out, and here they branch out as well. And so there's a bunch of places on the neuron that these are sort of right next to each other, and they transmit neurotransmitters, right? Which basically tells the neuron, should I be trying to send a reaction or not? And if the neurotransmitters are, sent into the cell, right, it triggers an action potential, right? And so these are a little bit complicated. The idea that you need to know is it has something to do with sending ions in and out of this long part of the neuron called parts of called the axons, right? There's a like sort of a bunch of these little axon parts. And it sends ions, sodium and potassium ions, in and out of the cell, and that creates an electrochemical signal, right? And so you don't need to know the details of how it sends things in and out of the cell, but you should know you have these neurotransmitters that enter into the neuron, right, from one neuron to the next. It triggers an electrochemical signal through the exchange of sodium and potassium ions with proteins obviously helping them. So here's a great way, right? You could disrupt neurons. You could damage these proteins, or, right, this process is facilitated by proteins. There are a lot of things going on at this process. If you could stop things from getting from one neuron to the next, right, that's going to be one of our main critical steps, one of the places things can go wrong. Indeed, the AP exam last year asked a question very similar, very, very similar to the question that you will see in question 2 of this AP exam. This is a good place to pay attention. So let's read this question. Also, I want to show you this picture of an action potential. You're at, negative 70 millivolts, and the action potential goes up. You have a big voltage. It goes up to about 30 millivolts, and it goes back down. It sort of overshoots and brings back. The threshold is sort of just if you get to negative 55 millivolts, if you get an electric potential like that, it's sort of the point of no return, right? Once you induce that kind of potential, everything starts happening, right? Now, it's important to know, and you'll see why, that these action potentials always go from negative 70 to about negative 30 and then downshoot a little bit, or negative 70 to positive 30 and then overshoot a little bit on its way back down, right? That's always going to be the same. It doesn't matter how strong the signal is. The signals that your neurons send each other, everything that you think, everything that goes on that you tell your body to do, right, is just based on how frequent the action potentials are, what frequency you send the action potentials with, not their amplitude. Their amplitude will always be the same. So let's go answer this question. Acetylcholine is a neurotransmitter that can activate an action potential in a postsynaptic neuron, right? So that's just the synapse is the space between the neuron, right? So presynaptic is where the signal comes from. Postsynaptic is it receives the signal, right? So shown in these figures. A researcher is investigating the effect of a particular neurotoxin that causes the amount of acetylcholine released from presynaptic neurons, right? It affects the presynaptic neurons. So let's see, it affects the presynaptic neurons to increase, right? It causes the amount of the neurotransmitters to increase. So let's look at what the questions are. Describe the immediate effect of the neurotoxin on the number of action potentials in a postsynaptic neuron. Okay, so if it increases the amount of neurotransmitters that get released, right, what will that do to the action potentials? Well, the neurotransmitters tell the next neuron, fire these action potentials. That's what it signal. So part A, let's start. It will increase. That's the first part. So this is our description, right? So you'll want to say, if it's asking you to describe it, we'll say, the number of action potentials increases. Okay. So then next we want to predict. So predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. Well, I just told you that the maximum potential, this amplitude, doesn't change, so it will stay the same. Right? Everything is based on that frequency, that number in any given point in time of the action potentials. Okay, the researcher proposes two models A and B for using acetylcholinesterase, an enzyme that degrades acetylcholine to prevent the effect of the neurotoxin. In model A, ACHE is added to the synapse. Okay, so model A, we're going to add the enzyme there, right? So in model A, it's added to the synapse. In model B, it's added to the cytoplasm of the postsynaptic cell. Okay, what's going to happen? So let's think about it. It makes the neurotransmitters go, more. It makes you have more neurotransmitters. Think about, right? Does it matter what's going on inside the postsynaptic neuron? Right? Once the neuron gets the neurotransmitter, it fires off these action potentials. So stopping the process, stopping the neurotransmitters after they've already stopping the neurotransmitters after they've already signaled a reaction, B is not going to do anything. Right? No, it's not going to do anything. A, right? If we stop the neurotransmitters in the synapse before they get to that postsynaptic neuron, right, that will stop the signal, right? The signal is right when these neurotransmitters get to the postsynaptic neuron, right? So model A will work, right? It will make everything normal. It will really stop the action potentials, right? Because it will get rid of those neurotransmitters before they can really reach the postsynaptic neuron. Once they've reached the postsynaptic neuron, stuff starts happening, so you've got to stop it before. So that's why model B doesn't do anything, but model A will work. Right? So this is a process with a disruption, and then the AP wants you to analyze that disruption.